Hey learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we are going to study about what is gene. Genes are the basic unit of heredity. When you talk about heredity, it means that traits which are passed from parents to offsprings. So genes, they contain the basic information of the traits that are supposed to pass from parents to their babies or offsprings. For example, hair color, eye color, height, or even disease resistance, or what kind of disease they are going to have in future. All the informations, they are stored in gene. So what actually gene is? Gene is a segment of DNA that determines the sequence of amino acid of a protein or RNA molecule. So inside a cell there is a nucleus present, and inside this nucleus DNA is present. But when the cell starts to divide, this DNA is condensed into a chromosome, and this chromosome is the site which contains gene, or genes, they are located on the chromosomes. When you unwind the chromosome, you will have the DNA, which is a double helix structure. Not all the DNA is a gene, but specific segment of DNA which contains specific nucleotide sequence is known as genes. So each gene is made up of specific sequence of nucleotide, for example A, T, G, C, G, A, or whatever sequence they have. The reason or the purpose behind is to encode a protein through the process of transcription. Transcription is a process in which the gene sequence is converted into an RNA. So once the RNA is here, this RNA will be translated into protein, and that protein in the body is going to show its effect, whether in the form of eye color, hair color, or in the form of some enzymes. So this is what gene is. Gene is a specific segment of DNA which contains instructions to form protein, and that protein is formed by the process of transcription and translation. When we come towards the components of gene, or when we see what gene is made up of in detail, or how this gene is working to form protein, then there are 6 important components or basic components of a gene. It starts with promoter. The promoter region is located at the beginning of a gene. So for example, we have this gene sequence, or you can say that this nucleotide sequence. When we zoom it in, then we have the start of the gene, and this is called as promoter. It contains the DNA sequence that signals the cell to start transcription, because we know that transcription is the first process of protein formation, in which this DNA is going to be converted into RNA. So it starts with the promoter. Like a promoter name indicate that it is going to promote, or it is going to initiate or start something. So the promoter is going to start the process of transcription. Then comes the exons. The exons, they are the coding sequences that contains the information to make protein. So exons are the specific regions which are going to form proteins. Here, for example, this purple part is exon. Again, we have exon, and then we have exon. So this part is going to make protein. Along with exons, we also have introns, as the name indicates that they are intermediate or in between the exons. These intron regions, they are non-coding sequences. It means that they are not going to make protein, but they are just present. The purpose of these introns is still a mystery. People are searching to find out why the introns are present in between the exons, but we don't have the clear information about it yet. So the introns are present in between two exons, and they are non-coding regions. They are not going to make any protein. Proteins, they are made by exons. Then comes the termination region. As the name indicates, it is going to be related to the stopping or termination of the transcription, and they are found at the end of the gene. It signals the RNA polymerase to stop transcription. And at the end, where the termination region is present, we have the stop codons. The stop codons, as the name indicates, they are going to stop the transcription, and they might be UAA, UAG, UGA in case of RNA, or TAA, TAG, TGA in case of DNA. So they are going to signal the termination of translation or transcription depending what kind of codons are present. If it's present in RNA, they are going to stop translation, but if it's present in DNA or gene, they are going to stop transcription. Along with this, we also have start codons, which are known as AUG. They are present at the beginning of the gene, and they are responsible for the start protein formation or RNA formation directly by the process of transcription. So that was all about gene. If you wanted to learn about more gene, or is there any other topic that you wanted to learn, just let me know in the comment section, and I will see you in the next video.